Hello everyone, welcome back to one more video session. In this session, I'll be speaking about another important concept, that is packaging of DNA. Could be asked for 1, 2 marks, 3 marks or 5 marks. Very important. First, I'll explain the packaging of DNA in eukaryotes, where you know that the DNA is present inside the nucleus, whereas in prokaryotes, nucleus is completely absent. Instead of that, the DNA is scattered within the cytoplasm. First of all, I am talking about in eukaryotes, how the DNA is packed. Why the DNA should be packed? The simple reason is, if you look into the structure of the nucleus, so this is the nucleus in eukaryotes, and this is actually the DNA. In other words, we can call it as chromatin thread. So this is the chromatin thread. Obviously, we know that in human being, if you just take a diploid cell, the total length of the DNA is about 2.2 meter. So that is, the DNA is of higher dimension. It's very lengthy. If you just take the 46 chromosomes, all the chromosomes are made up of the DNA. So if you just pull the DNA of all the chromosomes, it will make up 2.2 meters. It's very long. How this such a big DNA is present inside this nucleus? This is called as the packaging of DNA. So that means this entire DNA should be packed in a very good manner so that it will get fit within the nucleus. So this DNA is now available for the synthesis of proteins by a mechanism called as transcription. This is the mechanism. Got it? So this is all about the packaging of DNA. Got it? Okay. Now, how do you calculate the length of the DNA? By a simple formula, the length of the DNA can be calculated by just take the total number of base pairs in a DNA. Take the total number of base pairs if it is given in any such problems. Okay, what is the total number of base pairs are given? Then it should be multiplied with the distance between the two nitrogenous base pairs. What is the distance? 3.4 Armstrong units. That's it. So you just calculate it, you get the length of the DNA. This is it. So I have already written, it's huge, having high dimension. Compared to prokaryotes, eukaryotic DNA is very long, whereas prokaryotic DNA is very short. Okay, so that's the reason why it's scattered within the cytoplasm in prokaryotes. Now, to understand this, first what I'm going to do is the charge of the DNA. Let me just tell you, the DNA is negatively charged. Why it is negatively charged? Because of the presence of phosphoric acid. You know that a phosphate group is present in the DNA, which gives negative charge. The second thing is, we need to learn another type of component which helps in the packaging of DNA. They are a type of proteins called as histone proteins. You call them as histone proteins. All these histone proteins are positively charged. Please remember this, why they are positively charged. If you just draw a histone, let us imagine that histone is nothing but a protein, should be made up of amino acids. Okay, this is one amino acid. Simply, right? Okay, so whereas here they have the amino group that is having positive charge, that is in their side chains. One amino acid among many amino acids that forms the histone called as lysines. Lysines, they have positive charge. Another amino acid that is called as arginine. Arginine, remember it, lysines and arginines, they have positive charge. These are the amino acid which is present in the histone, which gives positive charge to the histone proteins. Remember it, you may get a question. So this is the thing. The next is, what are the different types of histone proteins? If you look into the histone protein, there are 5 different types of histone proteins are there. The first one is H1. It always occurs like it's always one. There is no pair for this H1 histone protein. This is H1. The other one is H2A. It always occurs in pairs. There are two H2A. Then H2B, that is always occur in pairs. Then H3 always occur in pairs. Then finally H4 always occur in pairs. So total there are 5 different types of histone proteins. H1, they are always one, whereas H2A, H2B, H3, H4, they always occur in pairs. And ultimately, these histone proteins, they are arranged in such a way that, for example, if I just consider this, so this is H1 protein. So this is H1 protein, which is normally outside a ball of histone octamer. Now I am going to draw the histone octamer. So this is one histone octamer protein, that is H2A. Let us consider this is H2A, this is H2B, this is H3, this is H4. The back side of this also contains the same thing. That means I cannot draw it, but still. So this is also H2A, and again somewhere else here, the behind H2B there is one more unit, that is H2B. That means all H3 or H4 occur in pairs. Ultimately, how many proteins are there? Histone proteins are there total? 8. So we call it as histone octamer. Please remember this, histone octamer. It is also called as, for your need, remember it, new body. You can also call histone octamer as new body. Okay. Now H1 is situated outside, whereas the histone octamer is bit like they are made up of 8 histone proteins. Okay, so this is called as histone octamer. This is H1. Remember. So this is the thing, the basic. Now let us imagine this is the DNA. DNA is very long. Correct. Okay, so DNA is very long. Now this DNA, when it comes in contact with the H1, so H1 is the first histone protein which brings the DNA into this histone octamer. That's it. It's like they help this DNA to be intact. So they bring it to the histone octamer. Now the DNA, see here, DNA wraps around this histone octamer. How many times now? One round is completed. Still again it continues further to complete its second round. But remember that it will not complete a full second round. Remember it. So again it comes in contact with the H1 and now leaves. See, it leaves. It goes like that. So this particular component, what you see is negatively charged DNA and positively charged histone octamer. These two components make nucleosome. Remember it. What you call nucleosome. You may get a question, what is nucleosome? Very simple. Negatively charged DNA wraps around the positively charged histone octamer with the help of H1 histone. Then it is called as nucleosome. Don't get confused with the nucleoside or nucleosome. Both are different. Got it? So this is the first technical term you have learned today. This is it. Now what happens to this DNA again? So because of the formation of the nucleosome, some sort of the length of the DNA is now reduced. Bit reduced, right? Reduced in the sense it is not, I should not say technically it is reduced, but you know that the length is now bit shortened. It is condensing. Next is again when it comes in contact with another histone octamer. Okay, so there are 8, and again what you see is another H1. H1 now brings this DNA to this histone octamer. Now that this DNA that wraps around the histone octamer will not complete the complete or full 2 terms. Instead of that, it will complete only 1 3/4 terms. Just remember this. Okay. So in histone octamer, you see 1 3/4 terms of the DNA. This is the thing. And now you may get a question like, how many nitrogenous bases are there in this DNA which wraps around the histone octamer? Remember it is 200 base pair DNA. So 200 base pair DNA now wraps around the histone octamer. This is the thing. Whereas what about this? The H1 which actually brings the incoming DNA to this particular histone octamer, and also it takes this DNA from the histone octamer to outside. This is the thing. So H1 protein or histone protein is the common kind of door for the entry of DNA, also exit of the DNA. This is the thing, H1. And again actually the same thing will happen with another thing. So this is another nucleosome. So in the nucleus you will find many millions of nucleosomes. And the distance between each nucleosome obviously occupied by the DNA. So this is the DNA, right? So between the two nucleosomes, what you see is the DNA. This DNA is called as linker DNA. Remember, you call it as linker DNA. It's very important. Linker DNA. Now you know that as the DNA is very long, obviously they will come across with the many nucleosomes or many histone octamers, obviously. So again this happens, it goes like that. So you will see millions of nucleosomes. All these nucleosomes are now called as solenoid. Many nucleosomes are called as solenoid. Remember it, very important. Solenoid. Many nucleosomes finally makes solenoid. Now many solenoids we will see at regular interval. For example, this is one nucleosome, this is another nucleosome, this is another, this is another. Finally, now you call this as solenoid. Again you will see another solenoid where you see kind of like a condensed DNA or like more number of nucleosomes at particular what you call stages or intervals. Fine. So this is the thing. So this is actually now together called as solenoid. Now many solenoids and ultimately called as, so these are all linked actually because it is one DNA and called as chromatin fibers or chromatin fibers. This is it. So this is the basic thing. Now what is important is, let me just use the board somewhere else here. Okay, I'll write here. So let us assume that this is now one solenoid. You see many nucleosomes inside. This is actually another solenoid. This is another solenoid. This is another solenoid. This is another solenoid. I mean, that is nothing but they are all linked. See. Okay. So this is the thing. Now at higher level, it requires a special type of protein called as non-histone. I'll write for non-histone chromosomal protein. I write here chromosomal proteins. I write here NHC, non-histone chromosomal proteins. They are not histones. They are different types of proteins, bigger proteins. They actually bring all these solenoids intact, making it it looks like a wheel. So like this. That's it. So this is it, and like this, and at the center you see a long thread. That thread is nothing but non-histone chromosomal protein. So this is actually the packaging. As a result, the DNA is now reduced in length or it is condensed. This is the thing. Okay, so this is the thing. Now I have a simple thing to tell you here. Okay, so let us imagine this is the DNA and this is now histone octamer, and my thumb is H1 protein. So this is it. So here what you see is when the DNA comes inside, first is it comes into contact with H1, wraps one turn, then again it wraps around. See, one turn is completed, it is going to complete the second turn, but it will not complete the full second turn. Now it leaves by just turning around or like wrapping around the H1, the same H1. This is it. So this is the thing. Got it? So it goes and meets another nucleosome. This component is called as nucleosome. What is it? Negatively charged DNA and histone octamer. Got it? Along with that H1 is there. What is the H1? What is exactly the other name of H1? These are the linkers. You may get a question. Name the histone protein which acts like a linker in nucleosome. The linker is H1. Don't get confused with the linker DNA. Linker DNA is different. That is the DNA present between two nucleosomes. That is actually the linker DNA. This is only linker. Got it? What is the function of this H1? Which keeps the DNA intact. That's it. It just hold the DNA tight. That's it. This is the thing. So this is all about the packaging of DNA. Now let us talk about what is exactly the packaging of DNA seen in prokaryotes. Simple. So this is the bacteria. Okay. So now I take one more chunk piece. This is now what you see is the DNA. Correct. And this DNA is with the help of a special type of non-histone proteins. Again I am telling you, don't get confused with this. This is non-histone chromosomal protein which is present only in eukaryotic cell along with the nucleosomes. In this case, they have a special type of protein which take the function of histone proteins. They are called as non-histone proteins. Even these histone proteins also positively charged. And here the DNA is negatively charged. That's it. What now this structure, that is negatively charged DNA, wraps around the non-histone protein in prokaryotes, making a structure called as nucleoid. Remember this, very important. Nucleoid. Don't get confused with the nucleotide, nucleoid, nucleosome, or what is it? This one, linker DNA. Many things are there. So nucleoid means it is seen only in prokaryotes when the negatively charged DNA wraps around the non-histone protein which is positively charged, making a structure called as nucleoid. This is packaging in DNA. This is all important. Got it? Okay. So now I am going to talk about euchromatin and heterochromatin. For that I need to erase the board. Fine. Now let us learn about two different types of chromatin which is seen within our DNA itself. One is called as euchromatin, the other one is heterochromatin. Euchromatin is nothing but, for example, if you look into the diagram, see this is a normal DNA and you could see here DNA. Now this is nucleosome, this is another nucleosome. I forgot to mention one thing, that between the two nucleosome, the DNA which now connects two different nucleosomes, this is one nucleosome, this is the second nucleosome, this is third nucleosome. So this DNA, which is now called as the linker DNA, as I said earlier, linker DNA, you may get a question like, what is the total number of base pairs in linker DNA? That is 80 base pairs. Whereas in this case, the DNA which wraps around the one nucleosome or which forms one nucleosome, or it could say like the DNA which wraps around the histone octamer, that is 200 base pairs. Please remember it, very important for need. Okay, so this is linker DNA, the linker DNA is 80 base pairs, like that it goes. Fine. So here, so now you are seeing that this is now simple becoming bit complex, and here you could see more complex structure. That means you could see more histone octamer, and you see many nucleosomes, that is solenoids are there. This is solenoid now. This is now one solenoid. So like that you see millions of solenoids. Now, what is this euchromatin? Euchromatin is nothing but a particular structure of the chromatin which is transcriptionally active. So for example, see here, this is very complex. So many nucleosomes are there which are now actually very close to each other, and if you stain it, if you just use any color to stain it, normally like for chromosomal staining, there are so many staining methods are there. For example, chromosomal painting or Giemsa staining, like that. But when you stain it, if it stains dark, then that particular chromatin is called as heterochromatin. Normally heterochromatin stains dark. This is very important. Whereas somewhere else here you could see that here the solenoid is not complex as this type of solenoid, where you could see the DNA is bit exposed. Matlab like it is now available for some other molecules for the transcription process. So this particular piece of the solenoid is normally called as euchromatin, and when you stain it, it normally takes very less stain and it stains lighter. So what is heterochromatin? Euchromatin. First we will go with euchromatin. This is actually stains lighter and it is transcriptionally active. Please write this point. Euchromatin is transcriptionally active and it stains lighter. Heterochromatin is transcriptionally inactive. What is this meaning of transcriptionally inactive? And it stains darker. Transcriptionally inactive, very simple, like this particular structure is very complex. Obviously the molecules like RNA polymerase, DNA polymerase, all these things cannot take the information from this DNA because the DNA is now congested, it's highly condensed. In this case it is not condensed. So wherever, for example, like a polymerase enzyme can come and then actually take the information, but in this case it's very difficult. So this is euchromatin, which is transcriptionally active site. This one is transcriptionally inactive. It cannot produce any proteins. This particular heterochromatins cannot be a gene, whereas euchromatins obviously gene. Now it's too early to say what is a gene and all this stuff, but I'm just giving you the idea for future classes. Got it? So this is the thing. So that is the difference between eu and heterochromatin. Remember it, very important. You need to write 2 points for both. Now this is actually solenoid. You could see here, from here the solenoid goes. Got it? So there are so many things here, I have not mentioned it, but it's understood. Okay, so fine. So these are all the solenoids. And now I said like at higher level, it requires some complex proteins, that is higher proteins, to just package the DNA within the nucleus. That proteins are actually, I have just mentioned here, that is the proteins are non-histone chromosomal proteins, which is also seen in our body, that is within our nucleus. There are 3 types. One is scaffold proteins are there, functional NHC proteins, that is non-histone chromosomal proteins are also there, or high mobility group proteins. Now I will not discuss about it, it is also called as HMG. All these 3 higher proteins actually help. Particularly what you see is the scaffold protein here. These are all called as the scaffold proteins. These are all higher level NHC, that is non-histone chromosomal proteins, which now actually brings all the solenoids. You could see here, and now it's becoming more condensed. Actually, it's becoming very condensed. Like that's it. So the DNA length is now being shortened. And now again here, you could see here, see what has happened to it. It is now condensed, highly condensed. Okay, now see here. Finally, it has become a chromosome. That's it. So this is all about packaging of DNA. Ultimately, it becomes chromosome. So that's what, like, from higher to lower. How do you go? Chromosome. Take a small segment of it, zoom it. You see this. Again, zoom it. You see this. Again, zoom it. You see this. And finally, ultimately, at last, you see DNA. This is it. Got it? Hope you understood this. This is only for need. For board, it is not required. But these two are very, very important for board. Euchromatin, heterochromatin. That's what I'm telling you now. Learn it. Very important. Don't lose 2 marks. It's very easy. Got it?